Good morning, and you have tuned into Murmu Biology. Let's see, we were seeing Jharkhand eligibility test life science detailed solution question number 71. And question number 71 is a bit long, but the bigger the question is, the simpler it is. Okay, and the much smaller concept on a much smaller concept, and the literature given, the question is pretty simple if you go through it patiently. Okay, so let's go. The tDNA region of a tumor inducing plasmid of agrobacterium tumefaciens carries genes that alter plant hormone biosynthesis, leading to crown gall tumor formation. So this is about a agrobacterium tumefaciens, which is causing crown gall tumor formation in plant. And then second paragraph, in a mutant strain, disruption of gene X result in the formation of rooty tumors, whereas disruption of gene Y leads to the shoot proliferation within the tumor tissue. Okay, and within the tumor there is shoot formation and root formation. You know, shoot and root. Shoot is the upper part of the plant and root is the lower part of the plant inside the soil, based on hormonal imbalance implied by these phenotypes means that is the outer appearance of the uh, organism. Which of the following statements correctly identifies the function of gene X and gene Y? So based on that data, the following options are given. Okay, so we have talked about agrobacterium tumefaciens in question number 61. You can go through it and have a pretty much clear idea which what it is, and we will have the introduction here also. So agrobacterium tumefaciens is a rod cell bacteria, gram negative bacteria. Okay, and as you know, in prokaryotes and in particularly in bacteria, there are two chromosomes, isn't it? One of them is plasmid and one of them is its own bacterial chromosome, isn't it? So for agrobacterium tumefaciens infects the plant for food, isn't it? And the plants get the its product, and the plants provides food to the bacteria, isn't it? So this is a kind of symbiotic kind of relationship in between the uh, agrobacterium and the plant. And you can see here the plant cell and the agrobacterium has a particular relationship, and in particular exchange is not a particularly correct term. The agrobacterium sends its tDNA into the nucleus of the plant to get integrated into the plant genome, and the plant produces cytokinins, auxin and opines, from which the cytokinins and auxin are utilized by the plant itself, and the opine is produced for the agrobacterium for its, which is the food of the agrobacterium, the opines which are the food of the agrobacterium, isn't it? Again, this is the same similar figure. The agrobacterium, the tDNA is transferred into the wounded site, through the wounded site inside the in the wounded cell, in the plant cell, and in then the integration of the tDNA, that is tDNA from the agrobacterium, makes the uh, plant to makes the plant food for the agrobacterium, that is opines, isn't it? So now you have seen that agrobacterium has two type of chromosome. One is its own chromosome, and one of it is the tDNA, ti plasmid, that is. And inside the ti plasmid there is tDNA, and you can see here, just pay attention, there are genes for auxin and genes for cytokinin, isn't it? And genes for opine also. Okay, so this auxin and cytokinin, just you remember these two terms. Okay, it will be helpful for you in the upcoming questions. So totipotency is a term through which a whole organism can be produced from the single cell, isn't it? And in plant, all the cells, each and every cell is totipotent, that is, each and every uh, cell of the plant can produce a new plant similar to its parent plant, isn't it? But in human beings or in other animals, all the cells are not totipotent. Only the zygote and up to maybe up to 8 cells it is they are totipotent. That means if you differentiate differently, if you culture differently in these 8 cells, then 8 organism can be produced. Okay, so that is known as totipotent. So using that property, that each cell of the plant is totipotent, the whole biotechnology of tissue culture is plant tissue culture is based upon, isn't it? And in plant tissue culture, what do we do? We take one cell of the plant, or better, explant of a explant is nothing but a part of the plant. It can be shoot, it can be root, and we put the explant in a culture medium. It is culture medium is what it is. Culture medium, it is the nutrient liquid broth in which the plant grows, isn't it? And when you give hormonal treatment like hormones like auxin and cytokinin and gibberellin, the plant then grows into the full plant, isn't it? So this is a rough idea of the tissue culture. You already know these things. You can also see this in question number 66, plant tissue culture. I have explained there. Okay, now coming on to the concept of the question. Okay, question number 71. So in here you can see in tissue culture, the when the explant is placed in the uh, nutrient medium, there is a formation of a callus, isn't it? For group of cell, that group of cell is known as initial callus, isn't it? So that callus, if treated with high auxin and low cytokinin, that is, the auxin to cytokinin ratio is greater than 1. Okay, then there is initial root formation. First the root is formed, then afterwards the shoot will be formed. And again, if the callus is treated with high cytokinin and low auxin, that is, auxin to cytokinin ratio is smaller than 1, then the shoot is formed, and afterwards the root will be formed. Okay, you must remember this. Okay, when the high auxin and low cytokinin is present, then what it is formed, initially the root is formed and then the shoot will be formed. And in case of low auxin is present and high cytokinin is given, then there is initially shoot development, and then afterward in the future there will be root development. Okay, so when you have seen that in introduction of hormones gives production of shoot and root, isn't it? So if we infect the callus with agrobacterium tumefaciens, then also the root and shoot formation appears, isn't it? So you must keep this chart in your mind. Okay, and this is just a repetition. When auxin and cytokinin ratio is greater than 1, that is, auxin is more and cytokinin is less, then there is root formation first, and afterward in the future there will be shoot formation. And when the auxin and cytokinin ratio is greater than 1, smaller than 1, that is, cytokinin is more and auxin is less, then there is shoot formation first. Okay, and when auxin and cytokinin ratio is 1, then the callus remain undifferentiated. Okay, so in relation to the question, there are mutants, isn't it? Mutants, there is inactivation of the gene or a non-working gene. So in case of an auxin mutant, okay, auxin mutant means the auxin is not functioning. Okay, then in case of auxin mutant, if the auxin is not functioning, then what will be in effect? Cytokinin will be in effect, then there will be shoot formation. Okay, and in case of cytokinin mutant, then cytokinin is cytokinin is not expressing in what? In agrobacterium tumefaciens, then there will be root formation. You must remember this, and this is the data, okay, that you should remember. Now coming on to the question, option number so the question says what is the question? Let's read the question. In a mutant strand, disruption of gene X result in the formation of rooty tumors. See, in second paragraph, disruption of gene X result in the formation of rooty tumors. Okay, so the root is formed. So when the root is formed, in case of the gene X, isn't it? Gene X, when it is mutated, then there is root formation. Okay, and whereas disruption of gene Y leads to shoot proliferation. Again, gene Y, when it is formed, when gene Y is mutated, then there is shoot formation, isn't it? So just we have previously read, when in case of root formation is there, when root formation is there, then the mutant is cytokinin, and when the shoot formation is there, then the mutant is auxin is mutated. Okay, so coming to the next part of the question, what the next part says: based on the hormonal imbalance applies in the phenotype, which of the following statement correctly identifies the function of gene X? Okay, so let's read the question. Options: gene X encodes enzyme involved in an auxin biosynthesis. So you can see here, gene X is not involved in auxin synthesis, but gene X is involved in cytokinin synthesis, isn't it? That is being mutated. Okay, so this is wrong, and option B says gene X encodes enzyme involved in cytokinin biosynthesis. Okay, so now you see here, gene X was involved in cytokinin when it was mutated. Then in gene X, the root formation is there. And again, cytokinin mutant meaning only auxin is expressed. Okay, so here you can see gene X is encoding what it is encoding: cytokinin biosynthesis, and it is being mutated, isn't it? In earlier it has already been told that in a mutant strand. Okay, and gene Y encodes enzyme involved in auxin synthesis. Gene Y is involved in auxin synthesis, but that is being mutated. Okay, so there is a formation of shoot. And option C says both gene X and gene Y encode enzymes involved in auxin biosynthesis. This is not similar to the paragraph. And again, gene X encodes opine synthesis. This is out of the relevant point, isn't it? So as you can see, you must remember this. These are the this is the fact. Okay, this is fact based on the theory, and this is the chart which is in relation to the question in the what the atmosphere question has provided. Okay, so based upon this, option number B is correct. Okay, thank you, and you can pay ₹1 for these tutorials. Thank you, and stay tuned, subscribe, and like this video, share this video, download for offline viewing, and thank you. Stay tuned.